Hello everybody, I am Dr. Azmi Mohsin, Professor of Anatomy, and I welcome you all to Dr. Sam's Anatomy classes. In this presentation, I am going to teach you about retina. A retina, is the innermost layer of the eyeball. It's a highly specialized photosensitive layer. I believe you have this idea about the basic structure of eyeball. It has three layers from outside to inside. Outside is the sclera, the fibrous layer. Then the middle one is the vascular and pigmented layer. And the innermost layer is the retina. Okay, so this is a photosensitive layer. So when you looking at a distance, there are parallel beam of light which strike your cornea, transparent. This entire the pathway of the light up till it reaches the retina, it's all completely transparent layers. Firstly, it strikes the cornea, then the anterior chamber, then passes through the pupil in posterior chamber, then it strikes the eye lens, and eye lens is a biconvex lens. So it converges those beam of light such that the beam of light, when they strike onto the retina, the image formed is a miniature and an inverted image. Now, this visual stimulus, when it strikes the rods and cones, it converts this energy into a electrochemical stimulus or electrochemical impulse, which is carried along the length of the neurons in the form of an action potential. And this reaches via the visual pathway and it ultimately reaches to the cerebral cortex, where with the help of this pathway, now you are able to see the erect image and of real sizes. And not only that, you are also able to see the three-dimensional, the depth of the objects. I'll tell you in later of my subsequent lectures, because this entire visual pathway I'll be completing in part lectures. So this is the first lecture in my sequence of this optic pathway. So let's talk about retina. Here you seeing is a longitudinal cut section of retina. So let's get familiar with all the layers of the retina. Conventionally it's divided into 10 layers, and before that let me tell you, this is the eyeball you're seeing here. So this portion here, it has been cut out and been zoomed, and as I told you, the innermost layer is the retina. This much all is retina, and outside you see is the capillary plexus, that's the choroid, and the last most layer of course will be the sclera. Okay, so we'll talk about the layers of the retina. I believe it is visible to you, or should I zoom it a bit? Okay, now it's a little better. So look here, when we conventionally, as I said, it's made up of 10 layers. So we count that the convention is that the counting should start from the outer layer to inside. So from the outside you're seeing is this, can you see this? This is the pigment cell layer, right? This pigment cell layer on outside, it is meant to absorb the light which is coming from outside, and so it doesn't reflect back. So it basically absorbs the light. But while the light enters and reaches up to this, the first layer, it is actually passing through all these neurons. So the next layer, you can say the light falls from this order, like 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. So that's how the light is approaching towards the pigment layer. So here you find is the next layer, the second layer. The second layer you're seeing is made up of rods and cones. So these rods and cones are actually, you can see, is the peripheral modifications of these cells. Now these are the cell bodies of the rods and cones. These cell bodies of the rods and cones, they are actually forming the outer nuclear layer, which is the fourth layer, right? So these cells, these are the cell bodies of the rods and cones. They have their peripheral processes which are modified photoreceptors, and they are called rods and cones. But between the rods and between the peripheral processes and the cell bodies of these receptor cells, there is, you can see, a blue color line, and that is called the outer limiting membrane, right? So that will be the third layer. Okay, and now the fourth layer is this one. These are the cell bodies of the rods and cones, and rods and cones are nothing but are modified peripheral processes of these cells of the outer nuclear layer. Now these cells will have their axons directed inside, and these axons will, impinge with the dendrites of the next cells, and those are the bipolar cells. So these axons of the rods and cones, they impinge and they synapse with the bipolar cells, and there are other cells. Can you see the blue color cells here? They are the horizontal cells. So this layer, that's the layer of the axons of the rods and cones, they form the outer plexiform layer. This is the outer plexiform layer, right? And then you find in another cell layer which is made up of cell bodies of these bipolar neurons. And these bipolar neurons are considered to be the first order sensory neurons in the visual pathway. So these are bipolar neurons. They have the dendrites facing peripherally which synapse with the axons of the rods and cones, as well as there will be horizontal or transverse anastomosis with the help of these horizontal cells. Now this layer is called the inner nuclear layer, the layer of the cell bodies of bipolar neurons. Hold on. Remember, in the olfactory pathway, I had taught you before, there are already videos uploaded. You can see the peculiarity about the olfactory pathway was there the first order neurons were themselves behaving like receptors. The peripheral processes of the first order olfactory neurons were behaving as the olfactory receptors. The free nerve endings, the dendritic processes in the nasal mucosa, they were behaving as receptors. So that was a peculiarity about olfactory pathway. The first order neurons and the receptors were the same. Here in optic or visual pathway, these cells we don't count as the first order neurons. So this is called the layer of photoreceptors, means it rods, the three layers together. That means the rods and cones, the cell bodies of the rods and cones, and then the inner plexus, that's the axons of the rods and cones. So all this is called the layer of photoreceptors. We don't call it as the first order neurons. Clear? Okay. Then this layer is the inner nuclear layer. And obviously, if it's the inner nuclear layer, these also will have their axons directed inside. So the axons of the bipolar cells will synapse with the other nuclear layer present deep inside, and that is the ganglion cell layers, right? So the axons of the bipolar cells and synapse with the dendrites of the ganglion cell layer. And here they also have a transverse or horizontal anastomosis, and that is being done by the amacrine cells. So this layer is now the fourth and fifth. Now this is the sixth layer. This is the inner nuclear layer called the sixth layer. Before I study further, let me tell you that amacrine and the horizontal cells, if they ask where in actually which layer you will find them, so it is this first, second, third, fourth, fifth and sixth layer. So it's the sixth layer. The inner nuclear layer is the sixth layer, which includes the cell bodies of bipolar neurons as well as the horizontal and amacrine cells. They are also a part of this inner nuclear layer. And the axons of these bipolar cells, they form the inner plexiform layer, right? So this becomes the seventh layer. Now the eighth layer is the layer of ganglion cells. Now these are the ganglion cells. The ganglion cells will be the second order neurons, right? So the ganglion cells will also have their peripheral processes, right? So this becomes the eighth layer. The ganglion cell layer becomes the eighth layer. Now the axons of these ganglion cells, you can see they converge towards the optic, the green color thing here is optic disc. That's the point of exit of the nerve fibers. So the axons of these ganglion cells, they're actually forming, they're bundling up and converging towards the optic disc to exit out of the eyeball in the form of optic nerve. So if it's asked like what actually is optic nerve, so it is the continuation of the axons of the second order neurons of the visual pathway, and those are ganglion cells. Then can you see there's an artery entering here? There's a central artery of retina. So remember that the central retina of artery will of course will have different branches here, and they form capillary meshwork here, and this will be embedded here if it's asked where actually this central artery, the branches of the central artery of retina, where they're running. So they are also running here over to this ninth layer, right? They're running over to this ninth layer. And after all of this, there is a transparent thin sheet placed here on the innermost surface of the retina, which separates it from the vitreous chamber, which separates it from the vitreous body, and that is called the inner limiting membrane, which is the 10th layer. Got it? I hope you've understood. I have described it in detail. Now look here, important is, I believe I have zoomed it out enough, and can you see this ganglion cells? Important is why I have zoomed it. I'll tell you, there's a representation of the image which you are seeing from the field of vision. Obviously the light rays, they are straight, so the the rays coming from the upper visual field, they will fall down on the lower half of the retina. The light rays coming from the lower half of the visual field, they will strike on the upper half of the retina. This light rays coming from the temporal field will strike on the nasal field of retina. The light rays coming from the nasal field will strike on the temporal portion of the retina. So that's how the image is represented in the retina, that is inverted as well as a mirror image. So that has to be you can say ki somatotropic representation, or the representation of the image on the retina, has to be transmitted the same way along the optic nerve, optic tract, optic radiation reaching to the visual cortex. So what I want to say is there is a fixed pattern of these fibers to be conveyed in the optic nerve, and that is the retina. If you say like a perpendicular, a ray of light passing through the center of anterior and posterior capsule of the eye lens that reaches straight back, strike on the center or the posterior pole of the eyeball, and there it's this area of the retina that's called the macula, and in this macula there is further smaller point, and that's called fovea centralis. So these areas are the area of bright and clear vision. So these fibers, the macular fibers, they run in the center of the optic nerve, while the fibers from the peripheral retina, they run in the periphery of the optic nerve. Now, can you imagine like fibers from the center of the retina should pass actually into the center of the optic nerve, and fibers from the periphery of the retina should pass into the periphery of the retina. How will this happen? Okay. So, can you see this? This is the way how the fibers closer to the optic disc, you can see they are first of all traversing the entire length of this 9th layer, that's the layer of the nerve fibers, and it becomes more closer to the vitreous side, and then it reaches on the medial side, right? So the peripheral fibers will be the on outside, while macular fibers will be on inside towards the vitreous. Then only they can become centrally placed in the optic nerve. Okay, so that was a detailed explanation about the layers of the retina. Now look here how they are arranged, and this is how it is, the layers of the retina. Let's have a fast revision here. I mean, if ever in a section they ask you about counting the layers, right? So how will you think about, let's imagine there is no marking down here, they simply give an image, then ask about like tag, identify this layer, what is this layer and all. So to start counting one thing, you should have this idea that, as I told you, the outer layer is adjacent to the choroid, right? And the inner layer is adjacent to the vitreous. So that means the inner layer will be smooth, clear cut margin, because inside it is the vitreous. There's no connective tissue, there's no adhesions, no attachments there. It's a clean, clear cut margin. So that forms the inner limiting membrane. That will help you identify. Then the outer thing will be, having a lot of connective tissue attached. Maybe the vessel wall, the connective tissue of the choroid and all. So this will not be a clear cut margin, because, so this, remember that this on the outer side, you should start counting on the outer side, because the inner layer is smooth. Okay. Now look here, as I told you, there's a choroid here. They have shown is the choroid, and then between the choroid and the pigment epithelium, that's the first layer of retina, there is a specialized fibroelastic membrane, and that's called Bruch's membrane. So remember about Bruch's membrane, I will teach you in detail further. Right now just remember the pattern. So between the outermost of the layer of the retina, that is pigment epithelium and choroid, there is a fibroelastic layer that is called Bruch's membrane. Now see, this is the pigment epithelium. I told you the purpose is to absorb the light, and so that it does not reflect and get scattered. It actually is like a dumping ground where all the lights get converge and they get absorbed in the pigment epithelium. Then this is the layer, you can see this much is the layer of processes of photoreceptor cells. Processes of the photoreceptor cells are actually the receptors, and these are the peripheral process, and they are either the rods or the cones. Let me tell you that now these rods and cones are not homogeneously placed. In the retina, towards the center of the retina, like the opposite, nearer to the opposite, posterior pole of the eyeball, there is the area of more clarity of vision, right? And so that area has more of cones, and the peripherally placed retina has more of rods. So about these receptors, now let me tell you in detail that the cones actually are meant to appreciate the bright light and the colors in the light, especially red, blue, green. So they are meant to appreciate. And further, if you go in the detailed anatomy about this layer, you find that even the cones are specialized for different colors of light. There's special cones for absorption of special wavelength of light, and that's how you can appreciate the different shades of colors. Okay, and the rods are basically meant to appreciate the twilight, the dim light, or toilet light, when there is completely when you enter some dark space, and you can still appreciate few things. That's like because of the rods. So rods are, of course, they are meant for black and white vision and for the dim lights, to appreciate the visual impulses in the dim light. But that was about the characteristics about these receptors. But now about the count in in the retina. The rods are around 100 to 125 millions. Okay, while cones are around 7 millions. ठीक है, समझ में आया? ये डिफरेंसेस धीरे धीरे करके याद रखने की कोशिश करो तो चीजें याद रहती हैं. And let's when you talk about these numerical digits, we should it's preferable to recollect the past few information that I have given. How many we are stopping it here, we are stuck to this point. We'll start again with this. In between I'm revising few certain facts. Remember when I started this neuroanatomy series, the introduction of neuroanatomy, there I taught you about neurons, the number of neurons present in human body. Although it's a vague type of question, but still you might come across sometimes if they ask like, how many neurons do you have in a human body, in an adult human body? So remember, it is around 200 to 300 trillions. 200 to 300 trillions, right? So tell me how many zeros you will have in a trillion. 1000 has 3, million has 6. No, billions and not trillions, billions. 200 to 300 billion neurons you have in a human body. So let's see how many zeros you'll have in the figure. You have 3 for 1000, then 6 for million, then 9 for billion. And if you say 200 billion, that makes 9, 10, 11. So 10 to the power of 11. That is the number of neurons. This is the number of, this is the number of neurons. Next, remember what I was teaching you is about, olfactory nerve. How many receptor neurons, the first order neurons, you can say how many neurons, olfactory neurons you find in an olfactory epithelium? Anybody remembers? The answer is around 16 to 20 million. 16 to 20 million. 16 to 20 million. Hold on, this is, I'm not writing here. Actually, the space is less. Yes, 16 to 20 million. Instead of that, I should write 10 to the power of 6. That will be better. Into 10 to the power of 6. That is olfactory neurons. This is 10 to the power of 11 is the into 2 कर सकते हो. 2 to 3 होते हैं. 200 to 300 बिलियन न्यूरॉन्स होते हैं बॉडी में. और olfactory receptors कितने होते हैं? The first order neurons in the olfactory mucosa, they are around 16 to 20 million neurons. Okay, then अभी बात आई थी, किसकी बात आई थी? Yes, so we are talking is about the rods. Rods, I said, is around 100 to 125. 100 to 125 million है ना, तो 10 to power of 6 rods हैं. Okay, and just now I told you that 7 million, 7 into 10 to power of 6, that is the number of cones. ठीक है, that is the number of cones. One more thing is, like, how many neurons, the second order neurons, they are traveling in the optic nerve, here in the optic nerve. So that is also around 100 million. No, hold on. Yes, this is 1 million. 1 million. 1 million के लिए you'll write 1 into 10 to power of 6. That is the number of nerve fibers in optic nerve. Okay, so these are numerical figures. They sometimes placed in options in your MCQs. So, in between, whenever I keep on, revising it for you, so that you also get this information repeated every time. Okay, so, so I was talking about these rods and cones, right? So that you find that 7 million are cones in a single eyeball. This is, of course, in a single eyeball. I'm talking is single eyeball. But, and in single eyeball, you can imagine there are 100 to 120 million rods. So rods are much more in number than the cones. But now you think, even though rods are so many in number, but in the central retina, the cones are dominating. Means in the central retina, there are much of cones and less of rods. While in the peripheral retina, the rods dominate and there are hardly any cones in the peripheral retina. Similarly, in the center of the macula, that is fovea centralis, the side of clearest vision, there you have only cones. Got it? So, as you are focusing, reaching towards the center of the retina, your image becomes more and more clear. And as the images are formed on the periphery of retina, the images become more and more faint because of less capacity to convert the visual into, electrochemical, or you can say the less amount of cones there. And even the rods also keep diminishing in, number as they go towards the periphery. Okay, so that was about the photoreceptors. And these photoreceptors are actually the peripheral processes of these cells, and these are the cell bodies of the photoreceptors, right? So these are the nuclei of the, let me zoom it out. I think it's not zooming now. Anyways, so here you're seeing this is the layer of nuclei of photoreceptors. That's called outer nuclear layer. And in between, this is the outer limiting membrane. Outer limiting membrane, actually you can see outer limiting membrane demarks the limit of the receptors. So the first and second layer are outside the outer limiting membrane. And now, inside to this outer limiting membrane, you have this this is the outer nuclear layer. Now these nuclei of the rods and cones, they have their central processes, the axons, and that form the outer plexiform layer. These axons will anastomose with the dendrites of the cells of the inner nuclear layer. And this inner nuclear layer is made up of the bipolar neurons, or the first order sensory neuron. Remember this. And there are also, I told you, there are also transverse or the horizontal cells, right? And at this point, as on the inner side, there will be amacrine cells also. So, the cell bodies of the bipolar first order neurons, then the horizontal and the amacrine cells, all they are located here in the nuclei of you can say is the inner nuclear layer. Then the inner nuclear layer, the axons of these bipolar neurons, they form the inner plexiform layer. And this inner plexiform layer anastomoses, the synapses with the ganglion cell layers. This ganglion cell layer is the 8th layer. And these ganglion cells will also have their central processes, and the axons of the ganglion cells, they form layer by layer, and they converge towards the optic disc, and this layer is called the nerve fiber layer of the retina. Okay, and all this, and there will be blood vessels also here. Although blood vessels, they have shown it here also. Here he has shown is the blood vessels here also. So these are capillaries, cut sections of the bed capillaries, but actually the branches of the central artery of retina, they are running here in the nerve fiber layer, and from there arterioles, venules, and they all like capillaries, they reach and perfuse the you can say up till the inner nuclear layer, up till the bipolar cells. Okay, so up till the inner nuclear layer तक की perfusion होता है by the from the vitreous side. There are, I'll tell you about later on about the blood supply of retina. We'll discuss later on. Well, you have seen is the blood vessels, right? So that means the blood vessels also run in the nerve fiber layer, and on the inner surface, it is this thin transparent layer of nerve fiber, and this is formed by the let me tell you, there are some longitudinal cells placed here. They have not shown in this diagram, but let me draw it. There are few cells which are like this type of cells. Yes, so these are the cells which are like these type of cells. So they're pillar type shaped cells. which extend along the length of the thickness of the retina, and they have their two processes on the two sides. So they are like pillars, and these are Muller cells, these are glial cells. All what I have taught you before was all neuronal apparatus of the retina. Now you seeing is the glial cells. Now this Muller cells, of course, they have various functions. One of the important functions is the mechanical support, like a pillar. So remember, it extends from the outer limiting membrane to the inner limiting membrane. Between, it's like pillars, so that mechanical advantage they give is preventing collapse of these neuronal cell layers. And they have the foot processes both the sides, and these foot processes, with the help of astrocytes, there of course the astrocytes also. So with the help of astrocytes, these processes, they form the inner limiting membrane, and this one of course is a transparent layer. This one also is a transparent layer, because the light ray has to pass through. Okay, so that's about. And these Muller cells, they also have function immunity as a form of immune cells, and they also help to synchronize, or you can say converge, the visual stimulus. They also have processes. They have so many horizontal, vertical processes. They also help in synapsing with the adjacent neuron, just like the horizontal and the other amacrine cells. So they also help to you can say streamline the visual stimulus. And they also, important function of Muller is they are forming sort of insulation to these nerve fibers, because here in the retina all these neurons are unmyelinated. Let me tell you that oligodendrocytes you will find outside the eyeball, right? So you don't find the myelination of neurons here. So they are all unmyelinated neurons. Even this nerve fiber layers, they are all unmyelinated. So these Muller cells, they help to provide insulation between the adjacent neurons. Okay. So I think that was about the retina. Now let's see the next slide.